Hello and welcome to Stem Cell Technologies webinar entitled Optimize the Flow Cytometry Crossmatch Assay, Save Time Without Compromising Sensitivity. My name is Mike Jones and I'm a product manager for cell isolation products here at Stem Cell. We're very excited to be joined today by Dr. Robert Liwski, Medical Director of the HLA Laboratory at Dalhousie University. Dr. Liwski will be familiar to many of you already. He is the author of numerous publications in the HLA field and has recently led a Canada-wide proficiency testing program for the flow cytometry crossmatch assay. Today, Dr. Liwski will be discussing the optimized flow crossmatch protocol that he and his colleagues have developed, which reduces the time needed for the procedure by more than half without compromising sensitivity. Before we pass things over to Dr. Liwski, our own principal scientist, Dr. Karina McQueen, will be providing an introduction to the cell isolation platforms that Stem Cell offers to facilitate highly sensitive flow cytometry crossmatch analyses. If you'd like to ask a question at any time, please use the question box located to the right of the slides. We'll address these in the Q&A session following the webinar, and any questions that we don't have time to answer during today's presentation, we'll address after the webinar in consultation with Dr. Liwski. I'd also like to remind everyone that continuing education credits are available for the webinar. Each registrant will receive 0.15 CE credits and electronic certificate of attendance from Stem Cell Technologies. If you're attending in a group, please just send me the names of those attending and I'll ensure that everyone receives CE credits. Okay, so thanks for joining us and I'll now hand you over to Karina for the presentation.
Welcome everyone. The presentation today will be on methods to standardize sample preparation for cleaner, more consistent flow crossmatch assays. I'm going to start off by briefly reviewing how the flow crossmatch works. Then I'll describe several different strategies available to isolate total lymphocytes for use in the flow crossmatch assay. I'll then turn the presentation over to Dr. Robert Liwski, who will tell us about his efforts to optimize the flow crossmatch. So as you know, the flow crossmatch is a standard technique for evaluating the compatibility of potential transplant recipients and donors. In the assay, donor lymphocytes are mixed with patient serum. If donor-specific antibodies are present in the serum, they will bind to the corresponding HLA antigen on the donor lymphocyte surface. This interaction is then detected through the addition of a FITC-conjugated anti-human globulin antibody, which will bind to any human antibody present on the T or B cells. And this interaction is then detected by passing the cells through a flow cytometer. Mean channel shifts are then determined by looking at the fluorescence difference between negative serum, positive serum, and the test serum. Now, if you're going to isolate lymphocytes for flow crossmatch analysis, you want a cell isolation system that gives clean cell preps, has good viability, and most importantly, gives low background fluorescence. We recommend using a negative selection approach to isolate your cells of interest, which in this case would typically be total lymphocytes, or your T and B cells combined. We have a number of different cell isolation platforms that can be used to isolate total lymphocytes, T cells, or B cells for use in the flow crossmatch. These include RosetteCep and CepMate, as well as EasyCep and RoboCep. Now, all of our reagents are particularly suited to HLA work as they're gentle on cells, so cells have excellent viability, they're quick, most isolations take less than an hour to perform, and best of all, our reagents can be used at room temperature or at 4 degrees, and they're stable even if you leave them out on the bench for an extended period of time. So the real advantage for an HLA lab is that you rarely have to throw out expired reagents, and you'll have to validate your reagents less often. The first isolation system I'm going to describe is RosetteSep, which is our non-magnetic cell separation system. RosetteSep works on any sample that contains red blood cells, including whole blood, cord blood, and bone marrow. It has several unique advantages in that it's a one-step procedure that doesn't require the use of any specialized equipment. So you don't need magnets, you don't need columns. All you need for RosetteSep is a centrifuge. And because RosetteSep is a negative selection procedure, the cells of interest are not labeled with antibody and are immediately available for use in any downstream application. How RosetteSep works is very simple. Your unwanted cells are cross-linked to red blood cells through tetrameric antibody complexes. And what you end up with are these rosettes of red cells with an unwanted cell in the middle. This increases the density of the unwanted cell, which causes them to settle out with the red blood cells when they're passed through a density medium. To use RosetteSep, you simply add the antibody cocktail to the blood and let it sit for 20 minutes. Then you perform a regular density separation. Your unwanted cells will pellet with the red blood cells, leaving your enriched cells to be carefully collected at the interface. We've recently developed CepMate, which is a centrifugation tube with a specialized insert designed to speed up density separations. It comes in two different sizes. There's CepMate 50, which can be used for processing between 4 and 17 mils of blood. And there's CepMate 15, which can be used to process up to 5 mils of blood. Now, CepMate can be used alone to isolate mononuclear cells, or it can be paired with RosetteSep to isolate specific cell populations like total lymphocytes. If you're using CepMate alone, you add your favorite density medium to the tube by pipetting it through a hole in the center of the insert. You then pipette the diluted blood sample quickly down the side of the tube onto the top of the insert and the density gradient medium. You then centrifuge the sample for 10 minutes with the brake on, and instead of carefully pipetting off your enriched cells, you simply pour the enriched cells into a new tube. Your red blood cells stay trapped under the insert. So in this way, you can isolate mononuclear cells in about 25 minutes, including your wash steps. Now, CepMate can also be paired with RosetteSep to isolate specific cell populations. And this is actually the method being used by Dr. Liwski's lab to isolate the total lymphocytes that they use in their flow crossmatch. So to isolate the lymphocytes, you simply add your antibody cocktail to the blood and let it incubate for 10 minutes. While your blood's incubating, you add your density medium to the tube, again by pipetting it through the hole in the center of the insert. You then dilute the blood, pipette it quickly down the side of the tube onto the insert, centrifuge for 10 minutes with the brake on. Your unwanted cells will pellet with the red cells under the insert, while the total lymphocytes can then be poured off into a clean tube. So using RosetteSep with CepMate allows you to isolate your total lymphocytes in about 35 minutes, including the wash steps. Now, EasySep is our magnetic cell separation system. With EasySep, target cells are specifically labeled with dextran-coated magnetic particles using a bispecific tetrameric antibody complex. These complexes recognize both the dextran-coated particle and the target surface antigen. Magnetically labeled cells are then separated from unlabeled cells by placing the sample into one of our specially designed magnets. EasySep can be automated with an instrument called RoboSep. RoboSep is a pipetting robot that automates both the cell labeling and the cell separation steps. It can isolate up to 4 independent samples simultaneously. It uses EasySep reagents and procedures and does not use columns. The main advantages of RoboSep for an HLA lab are it saves technician time, as 4 independent samples can be processed in about an hour with only 5 minutes of hands-on time. It avoids sample contamination, as each sample is handled by a unique set of disposable filter tips. And as all the steps are fully automated, there's minimal handling of potentially hazardous samples. So in summary, we have 2 main platforms that can be used to isolate total lymphocytes or T or B cells for use in the flow crossmatch assay. There's RosetSep, our density-based cell separation system, which can be used alone or in conjunction with SepMate to speed up the procedure. And there's EasySep, our magnetic cell separation system, which of course can be automated using RoboSep. Now, before we move on to Dr. Liwski's presentation, we have a quick poll for everyone to complete on screen. So I'm going to turn the microphone over to my colleague Mike, and he'll walk everyone through the questions. So one moment, please.
Thank you, Karina. We're just getting this poll set up here, so if you'll bear with us for just a second. Okay, the first question you should see appear on your screens momentarily, and you can then just check and submit your response. So the question is, which method is used for flow crossmatch in your lab? The tube method or the 96-well tray method? We'll just give a few seconds for these votes to come in. Okay, it's looking like about 2/3 are using the tube method. Okay, thanks for that. And there's just one other question, which is, do you use pronase treated cells in flow crossmatch, yes or no? Okay, looks like most people are using pronase treated cells. Thank you. We'll have a couple more poll questions later on, but right now I would like to pass things over to Dr. Liwski, our guest panelist, for his portion of the presentation.
Mike. I would like to thank Stem Cell Technologies for inviting me and giving me the opportunity to share our experience with optimization of flow cytometry crossmatch. So I'm hoping to quickly introduce the flow cytometry crossmatch, especially to the directors in the audience who don't actually perform the assay. Also talk a little bit about the development of the rapid optimized crossmatch assay, dubbed Halifax Protocol, and some experience from Canadian and Brazilian proficiency testing, and then talk briefly about the new protocol that we've developed, which includes the rapid cell isolation technique that Dr. McQueen was talking about, using SepMate and RosetSep combo, and we call this a Halifax protocol. So we all know why we do cross-matches. Ever since this study that was published in 1969 New England Journal of Medicine by Dr. Paul Tarasaki and Patel, you see here that cytotoxicity cross-match was able to predict poor outcome or early rejection post-transplantation, whereas negative cross-match, people with negative cross-match had a relatively good outcome early on after transplantation. We've since improved the technology, and now probably 80% of labs in the US and most labs in Canada are using flow cytometric crossmatch. And in this assay, as Dr. McQueen described, we are using donor cells here and mix them with recipient serum, and then the donor-specific HLA antibodies bind the cells. It's important to wash the remaining antibodies off so that they don't interfere with the secondary reagents that we use for detection of positive or negative crossmatch. And this is your anti-IgG that's conjugated to a fluorescent dye FITC. This, of course, will bind the cells that are tagged with the antibody, and then we use the monoclonal anti-CD3 and CD19 antibodies, which are conjugated to different fluorescent dyes to actually be able to gate on T cells and B cells. And this is a typical gating strategy that is used by most laboratories. So here you have your CD19 positive B cells, CD3 positive T cells, and then we're asking question, are there antibodies tagging these cells? And here at the top is an example of a negative T cell crossmatch and B cell crossmatch. The reactions are below the threshold here in red, and every lab has to determine what this threshold is going to be based on a normal serum study. Here's an example of a weak positive crossmatch where you see the shift of fluorescence intensity due to binding of antibodies and FITC secondary reagent. And here's an example of a strong positive crossmatch. So this assay, as I mentioned, has been described back in 1989 by Dr. Gable and Bray, and this is the protocol that's from the ASHI manual. So the first incubation between the cells and serum occurs at 4 degrees for 30 minutes. Then there are 3 centrifugation steps to wash off the remaining antibodies that are not binding the cells, and then we incubate the cells with the secondary reagent and the monoclonal antibodies again for 30 minutes, followed by 2 more centrifugation and washing steps for a total assay time of 1 hour and 25 minutes. So this is not what you'd call a stat test. It takes quite a bit of time to perform. And there's really no evidence that these incubation times or the incubation temperatures, cell numbers, volumes, and wash times are actually optimal. Now, my background is from hematopathology, and as hematopathologists, we also run blood banks. And in blood bank world, there's a similar assay called the red cell crossmatch, and this assay takes only 25 to 30 minutes to get the results. And so I wondered whether we could actually optimize flow cytometry crossmatch and expedite the assay. And so we decided to ask this question. And so the objectives of our research was to develop an expedition flow crossmatch procedure without compromising the sensitivity of the assay. And we decided to investigate a number of factors, including the platform, so 96-well plate versus tube method, serum incubation time with the secondary reagent, the temperature of incubation, the number of target cells, and amount of serum. And here are 2 technologists, Jeff Adams and Jeff Paldo, from my lab that have performed majority of the experiments that I'll be describing momentarily. And here's the pooled positive control serum from about 20 highly sensitized patients that we use for a lot of the initial experiments. So the question between the, which assay platform to use, 96-well tray versus tube, there may be a lot of advantages to using 96-well tray because the centrifugation time can be reduced. You're spinning through much smaller volumes. And because you have 5 centrifugation steps, that could save a total of 20 minutes. Of course, during washing, flicking of the tray would be much faster than decanting supernatants from each tube individually. And you could also reduce the time that it takes the assay by speeding up the delivery of reagents and buffers using multi-channel pipetter when you're using the 96-well tray. And so here is Jeff Adams actually performing the tray method here. You can see how neat it is. You can quickly deliver reagents using multi-channel pipetter to multiple wells. When I asked him to repeat the same experiment in the tube method, he wasn't very happy with me. Here's the vacuum that we use to aspirate supernatants from tubes, and it takes quite a bit of time to do that. He was getting confused whether he already added sample to each tube, and he was falling asleep during the washing steps. And in the end, the results were identical. So here on the x-axis, you see our pooled positive control at different dilutions. And here you have delta channels from negative control serum. You see that there are two examples of cross-matches, T-cell cross-match and B-cell cross-match here. And you see that the results are virtually identical whether you use a tube method or plate method. But depending on how many samples you're running, there's approximately 30 to 40 minutes to be saved simply by switching from tube to the 96-well tray method. The next thing that we investigated was the effect of serum incubation time. So we performed a time course experiment. And initially, we incubated our positive control sera with cells for varying amounts of times, ranging from 3 minutes to 30 minutes. And then all groups were incubated with the secondary reagent, anti-IgG FITC, for 30 minutes. And here are the results. Again, just to orient you, on the x-axis, we have our positive control serum at different dilutions. And again, channel values, delta channels from negative control are on the y-axis. Here's where we would put our cutoff for a T-cell cross-match at 75 channels. What you see is that majority of the reactivity with serum occurs in the first 5 minutes of incubation. As you extend the incubation time, you're getting slightly higher reactivity, which plateaus by about 15 minutes of incubation. This was true for all different dilutions of positive control that we have tested. This is an example of a T-cell cross-match, and you see that the same results were generated when, for a B-cell cross-match. So next, we looked at the time course experiments, looking at the incubation with anti-IgG FITC. So what we did here is we incubated our cells and sera for 30 minutes, but then different groups were incubated with the secondary reagent for varying amounts of time, ranging from 5 minutes to 30 minutes. And the results were even more interesting, showing that you get maximal reactivity by 5 minutes of incubation. Again, this was true for all concentration of our positive control, and the B-cell crossmatch looked very similar. Next, we looked at the effects of incubation temperature, and what we found is that if we increase the incubation temperature from 4 degrees to the ambient temperature of about 25 degrees in our laboratory, hopefully, we are getting actually slightly stronger signal, again, at all concentrations of positive control tested, and the same results were obtained with the B-cell crossmatch. Next, we looked at the effects of serum volume to see if we could boost up sensitivity of the crossmatch by increasing the volume. The standard protocol that's described in ASHI manual calls for 1-to-1 serum to cell volume ratio. If you increase this to 2-to-1, you can see that you can increase crossmatch sensitivity. And again, when we did B-cell crossmatch, we found the same type of result. Next, we looked at the effects of cell number. So if you keep the amount of serum or antibody constant, but vary the amount of target, you may actually see differences in your results, and indeed you do. As you decrease the number of targets, you have more antibody available per target, you see that you are actually increasing sensitivity. This is 1 million cells per well, 500,000 cells per well, 250,000 cells per well, and 125,000 cells per well. And the results are quite dramatic. And in fact, here at the lower or high dilution of our positive control serum, you can see that it may make a difference between a positive and a negative cross-match. And again, we found similar results when we looked at the B cells. So in summary, there's a marginal effect of serum incubation time beyond 15 minutes and negligible effect of anti-IgG FITC incubation time beyond 5 minutes. Now, we can also increase cross-match sensitivity by increasing incubation temperature, increasing serum volume, or decreasing numbers of cells. And so based on all these results, we devised a shortened or rapid protocol, which we also call the Halifax Protocol. So you can see that the first incubation time is decreased to 20 minutes, second incubation time decreased to 10 minutes, all the washes are decreased to 1 minute per wash. And then to try to, because all these changes could potentially decrease sensitivity of the assay, we thought we would try to compensate for that by increasing the temperature of incubation to room temperature, increasing the volume of serum to 50 microliters, and decreasing the number of cells from 500,000 to 250,000. And so then we decided to perform a full validation of this assay using our negative control sera, predicted negative patient sera, positive control sera, as well as predicted positive patient sera based on Luminex assay. We want to determine our 3SD cutoffs. And so here are the results using the channel values versus MESA values, which some labs are using. So you have your T-cell and B-cell cross matches. You can see that with our negative control, pooled negative control serum, the two assays give virtually identical results. Looking at our predicted negative patients, we ran 90 patients in parallel between the two assays, and we ran another 50 predicted negative patients using the rapid protocol. And again, you see that the results overall are virtually identical. There were 2 false positive cross matches in each group, which is actually pretty good. And so the average positive cutoffs that we've been able to calculate based on these predicted negative patients were virtually identical for the two assays, the standard protocol and the rapid protocol. Our positive control serum is running slightly higher with the rapid protocol compared to the standard protocol. And these are the results of the predicted positive patients. Some of them were only predicted to have a positive B-cell crossmatch. Again, you see that in virtually all cases, the result interpretation is the same, and the channel values remain very similar between the two assays. So in conclusion, we can reduce the time that it takes to perform cross-matches without compromising the sensitivity of the assay, and this has some significant time and cost savings and perhaps impact on ischemia, especially if you're trying to perform cross-match on an imported donor, where the sample and the organ travel to the recipient center at the same time, and then the team has to wait for the cross-match before they actually perform the transplant. So this idea came up during my training. Dr. Gable is my mentor. We're discussing flow cross-match during one of our sessions, and we thought maybe we could speed it up. And Dr. Gable is always full of great advice. He always tells me to stay current because the HLA field is very competitive. At the same time as we were performing these experiments, in Canada, we started sharing organs. We have now 2 registries, the Living Donor Pair Exchange and the Highly Sensitized Patient Registry. And so we wanted to make sure that our testing across the country is actually similar. And so we decided to look at the degree of variability in the results in our flow cross-match assay. And to do that, we established a proficiency testing program. And we have representatives from each laboratory that on the proficiency testing committee. And so far, we have organized 3 proficiency testing surveys. And I'll talk a little bit about the first 2 surveys that we've organized. Here are all the laboratories and directors as well as supervisors from the labs that have participated. And I'd like to send special thanks to Denise Pochinko, who is the co-chair of the committee, for all her help and dedication, as well as 2 Jeffs in my lab who devote a lot of time to helping prepare these surveys. Now, the objectives of the surveys were to determine the extent of variability of flow cross-match results across Canada, and to try to identify factors that influence the results, and then to determine if standardization of flow cross-match could improve reproducibility of the results. And so in our first survey, we had all 13 labs participate. We sent 2 donor cells and 3 test sera to each lab, and we asked everyone for the result interpretation as well as raw channel data. And we also sent out a questionnaire asking some specific information about each protocol. And so here are the composite results, positive, negative, for in the first proficiency survey. You can see that overall the results are pretty good. Majority of the labs are in the consensus, although there are some outlying results. But when we looked at the raw data, we saw that there was a bit more variability than we expected. So again, to orient you here, on the x-axis, you have each serum, and the serum, we asked each lab to perform serial dilutions of each serum to make sure that they could titrate their cross-matches. And on the y-axis, again, you have your delta channels. Each line represents a different laboratory. So you see that there's quite a bit of variability in terms of how the channel shift from negative control for this particular cross-match. You also see a couple of labs having lower fluorescence intensity at when the serum is neat compared to diluted serum. And we later determined that this was due to poor washing technique, because there's a lot of unbound antibody here, which when serum is neat, that can interfere with the secondary reagent. What we also saw is that some labs, well, many labs have called this particular cross-match positive, B-cell cross-match positive. Other labs were calling it perhaps positive, but was very weak, and the cross-match titrated out very quickly. And what we noticed by correlating with the reported protocol from each laboratory is that these laboratories here were not using pronase to treat lymphocytes. And this wasn't just a characteristic of that particular cross-match. When we looked at another combination here, and this is now due to a class 2 donor-specific antibody, again, you can see that these labs that are not using pronase are detecting very weak or even negative B-cell cross-match, while the labs that are using pronase are clearly detecting positivity here in this particular cross-match. What we also noticed is that some labs exhibited a higher ratio, signal-to-noise ratio in a T-cell cross-match. And again, we're able to correlate with reviewing the protocol that these labs were using fewer target cells, 250,000, compared to the labs that had low signal-to-noise ratio in their cross-match. And these labs were using either 500,000 cells or more per reaction. So in conclusion, there was significant variability in cross-match results, and there were differences in protocols that we noted, and some of the factors that we were able to tease out appeared to be whether labs were using pronase for treatment of lymphocytes, the cell numbers that they use per reaction, and the serum volume, as well as washing technique. And so in the second survey, we wanted to see whether we could standardize cross-match protocol and whether this would improve the reproducibility of the results. And we also wanted to evaluate the performance of the Halifax protocol, the rapid tray method, nationally. And the reason for that was, and I want to draw your attention to it right now, is that the results from my lab, and this is the standard protocol, and in circle is the rapid protocol, were actually superior in terms of signal-to-noise ratio compared to other labs in the country, despite the fact that this is a rapid method. So in that second survey, we asked all the labs to perform cross-matches using their current lab method with the following modifications. We sent everyone our pronase and DNase as well as the protocol, so everyone had to treat cells with pronase. And we also standardized the number of cells and the cell suspension volume and serum volume. And as a second experiment, we asked everyone to repeat all these cross-matches using the Halifax protocol. And here we sent the instructions, the video to make sure that everybody was performing the technique the same way. And we provided some common cross-match reagents, including IgG FITC. And so here are the results. So you can see that by standardizing the numbers of cells and also by coaching some labs about the washing technique, we are now able to get the results to be more concordant. But when the labs repeated the same cross-match using the Halifax protocol, you can see that there was a better signal-to-noise ratio for virtually every single laboratory. And you can see that the concordance of the results also seems to be improved. And what I showed you was T-cell cross-match, and here are the same results for one representative B-cell cross-match. Overall, the signal-to-noise ratio was better for all cross-matches, T-cell cross-matches, when labs were using the Halifax protocol. The predicted negative cross-matches were still negative. And there was also some significant differences in some cross-matches. Looking at B-cell cross-match, some of them were not significantly different. When we looked at the precision, so how variable are the results, you can see that the result variability is reduced when all labs are using the same protocol, the Halifax protocol in this particular case. And you also see that overall, all labs had saved some time, even though they were performing this cross-match using the Halifax protocol for the first time. Overall, there was about a 1 hour time saved. So in conclusion, protocol reagent standardization can greatly improve precision of flow crossmatch. The Halifax protocol compared favorably with conventional lab crossmatch techniques in terms of improved signal-to-noise ratio, precision, and decreased turnaround time. And we are working towards the national implementation of this protocol in Canada. There are many labs across North America and Europe and other countries that are trying to adopt or at least evaluating the protocol. In fact, in Brazil, I was invited to help with the National Proficiency Testing Program to see if we could standardize the technique there as well. And so here are just some results that I want to share you. And again, I want to thank Dr. Jorge Neumann and Margaret Torres, past presidents of the Brazilian Histocompatibility Association, for inviting me to do this work with them. And so we've so far ran 2 cross-match proficiency testing surveys with 2 donor cells and 3 sera. And again, we simply asked the labs for cross-match interpretation and collected some raw data. Again, there's a reasonable concordance between each lab, although for some sera there are some outlying results. And here again is the raw data. This is one representative T cell and B cell cross-match. You can see that there's quite a bit of variability with some labs showing only about 50 channel shift for this particular cross-match, others about 370 channel shifts. And here's the Halifax protocol. So these are results from my lab. And also we asked how much time did it take for each laboratory to perform the actual cross-match technique, and you see that most of the labs took between 2 to 3 hours to perform this, and the Halifax protocol took only about 50 minutes. This is one lab, Dr. Jorge Neumann's lab, Porto Alegre laboratory. We actually compared our results side by side because Dr. Neumann's lab has adopted the Halifax protocol, and you see there was a great concordance between our laboratories in this particular proficiency testing. In fact, Dr. George Neumann has shared his validation data with me. They ran over 400 cross-matches in parallel between their standard protocol and the Halifax protocol, and you see that the correlation was excellent. There was only one false positive cross-match in each group, and it was just borderline above the cutoff false positive cross-match. Dr. Neumann's lab performs probably 10 times more cross-matches than my laboratory does, so he is the true world expert in performing the Halifax protocol. And he's spreading the word around. He's quite happy with this protocol, and I'm not sure, though, whether everybody is actually buying it. So next, I just wanted to talk about the Halifax protocol, where, as I mentioned before, we are using the rapid lymphocyte isolation technique. And again, I want to help Stem Cell Technologies for support with cell isolation reagents for our validation study. So we already talked about the fact that we can reduce the flow cytometric crossmatch time to about 35 minutes, but crossmatch assay is only half of the story, and cell isolation is, of course, another time-consuming event. And so here's a standard peripheral blood mononuclear cell isolation using a FICOL protocol. It takes 2 minutes to layer your cells, and 40 minutes spin without a break. And then you have to collect the layer, which takes quite a bit of time, and you have to be very careful not to contaminate your lymphocytes with monocytes and neutrophils. Then there are 2 washing steps, and of course then we treat cells with pronase and DNase, followed by another wash cycles for about an hour and a half. And this assay is, as I mentioned, or this cell isolation is time-consuming and gives you a variable purity depending on whether it's a deceased donor or live donor that you're isolating your cells from. And the purity ranges in my lab to between 30 and 80%. So here's the CepMate RosetteCep isolation protocol. As Dr. McQueen has described, there's quite a bit of time saving with the layering and the spin. Now, the cells that come out of this particular isolation technique, they have quite a bit or a little bit more red cell contamination. So we have introduced the ACK lysis step, and then we follow with the pronase and DNase. And so you see that the total isolation time with this particular technique is only 60 minutes. So we are saving 30 minutes by using this particular technique. And Dr. McQueen has already gone through the actual protocol using the CepMate and RosetteCep. but here's a quick glance at it again, and you can see a video of it at the Stem Cell Technologies website. So here is a representative result of our peripheral blood mononuclear cell isolation using standard FICO technique. You can see that there's a lot of impurities still present in this particular isolation, and lymphocytes are probably only about 25% here. And here is the same cell isolation using the CepMate RosetCep technique. You can see very pure lymphocyte population. We routinely get over 90% purity. Now the cell yield is decreased. We only get between 2.5 and 8 million cells from 2 tubes. And so because of that, we have to readjust our cross-match protocol, because if we are using 250,000 cells, sometimes you will not have enough cells to perform your cross-match. This reduction is possible because here you're dealing with unfractionated cells, where lymphocytes could only be, in some cases, 30%, as I mentioned before, whereas in the purified lymphocyte population with over 90% of cells, virtually all your cells are lymphocytes. So we were able to decrease the number of cells to 150,000, and we decreased the volumes of the lymphocytes and corresponding volumes of serum. Because we are using fewer cells, we are also decreased the amount of cocktail, which saves a little bit of money per reaction, and we decreased the secondary antibody incubation time to 5 minutes from 10 minutes. So now the total assay time is only 30 minutes, and I just quickly wanted to show you the preliminary results of our validation. So here's a comparison of the standard isolation technique versus RosetCep CepMate isolation technique, and this is a cross-match using negative control sera. So you see that virtually identical results. Here are predicted negative cross-matches. I think we ran 80, if I am not mistaken, and again, we didn't have any false positive reactions here, and you see very similar results between the two cell isolation protocols. Here are results looking at our pooled positive control sera. Again, there's virtually no difference between the two isolation protocols. And here are the results of predicted positive cross-matches. We used 16 patient sera that were predicted positive, and again, you see no difference in results interpretation, perhaps slightly higher channel shift when we are using the purified lymphocyte population. So in conclusion, I hope I've convinced you that we can reduce the time it takes to isolate cells, and the cells that you use are of improved purity, which make the analysis easier, and we can further reduce and optimize cross-match time, and this leads to even more time savings. And so here's a picture of my family in San Francisco during the 2009 ASHI conference, and we're taking these streetcars everywhere. They're quite reliable, but when you're in a rush, you probably want to take a bullet train. And I just quickly wanted to thank all my collaborators from the US, Canada, and from Brazil, all the people from my HLA laboratory who helped out with the project. And then, of course, the Canadian National Proficiency Testing Committee, Canadian Blood Service for sponsoring some of the studies that I described, the Brazilian Histocompatibility Association, all the laboratories that were involved in the proficiency testing. I can't thank them enough for their help. And, of course, the Stem Cell Technologies for invitation to share this, all this data with you. So I'd like to end here, and I would be happy to answer any questions. Thank you very much.
Great. Thank you, Rob, for that great presentation. Just as a reminder, if you have any questions for the Q&A session, please submit them using the question box to the right of the slides. And before we begin the Q&A session, we'd like to ask a couple more poll questions. So as before, you'll see the questions appear on your screen, and you can then just select your response. So if you'll bear with us for just a second. Okay, so the first question is, would you consider adopting the 96-well tray method for flow crossmatch? just give you a few seconds to complete the poll. Great, thank you. The next question is, would you consider using pronase-treated cells in flow crossmatch? Okay, that's great, thanks. And finally, would you consider using lymphocyte isolation for flow crossmatch? Excellent, thank you. The next question is, would you consider using pronase-treated cells in flow crossmatch? Okay, that's great, thanks. And finally, would you consider using lymphocyte isolation for flow crossmatch? Excellent. Okay, thanks very much. Excuse me for just a second here. Okay, we're now going to begin the Q&A session by addressing the questions that you've submitted. So the first question is for Dr. Liwski. What speeds, are the 1-minute spins you use?
Well, thank you very much for this question. The centrifugation speed is 500 G for the fast spins.
Okay.
And also I should say that anyone who would like to try the protocol, just please email me, and I'm happy to share the protocol with you.
Great. We had a couple of questions about that, so that's great. Thanks. Okay. Another question: are there any other benefits to using the tray method apart from the time saving?
Yes, of course. I mean, the big benefit is that you can run many samples at the same time. So, in my lab, we can do for example, a deceased donor, we would run our negative control serum in duplicate. We would run 5 different dilutions of positive control sera to make sure that we are titrating the cross-match properly. We would run, between 5 and 7 renal transplant candidates, 2 as the primary ones and 3 or 4 others as backups. And we would typically test at least 2 to 3 or sometimes 4 sera on each patient in duplicate. We would also run a liver transplant candidate, maybe 1 to 2 cardiac transplant candidates. Again, we would choose multiple sera on these patients, and that's all doable in a very reasonable time frame. On live donors, we can run, again, multiple sera. We could run the standard or the allo crossmatch and auto crossmatch at the same time. Sometimes, if we have 2 to 3 crossmatches at the same time, you could again run them simultaneously. We try to, or we have a policy that we run them on different plates. But again, you could spin up to 4 plates at the same time. So you could, once your reactions are set up, you can run multiple plates. So that really saves a lot of time and makes cross-matching possible.
Right. Great. Okay, another question for you here. Are the samples read in the tray or transferred to the BD tubes?
Yes, so if you have a attachment that can read plates, you could do that. And I know that the lab in Edmonton, Dr. Trish Campbell's lab, and Halpin is the supervisor there. They actually do that, and it seems to be working well. We don't have that attachment in my laboratory, so we do transfer from the plate into standard flow tubes. We can transfer using multi-channel pipetters, 3 tubes or 3 reactions at a time. So on a typical cross-match, if you have, let's say, 20 to 30 reactions, that would mean 6 to 10 transfers. So that takes only 30 seconds to a minute, and then you are on the machine.
Another question here. What is the ACK lysis? The questioner says they experience a considerable amount of RBC contamination with non-living donors in about a quarter of their isolations using RosetSep and SetMate.
So, sorry, Mike, could you repeat the question? Is it asking what is the ACK buffer, or what is the ACK buffer experience?
Both.
Okay, so the ACK buffer is ammonium chloride potassium buffer, and it's available through Stem Cell Technologies and other vendors. The experience for us is excellent in our validation. Even heavily contaminated red cell isolations, within 3 minutes, you perform your lysis and you see virtually no red cells at the end of it.
Okay, thank you. And the next question, would you recommend a 56C treatment of sera to inactivate complement before doing the cross-match?
That's a great question. It depends what your protocol, what sort of protocol you're using. And I think the main issue is the temperature. So if you run your cross-match at 4 degrees, a complement activation probably is not going to be a huge issue. And then, I don't think it's necessary to inactivate your sera. If you're running it at room temperature, you may get some activation, but this should only occur when you're predicting a strongly positive cross-match where you have complement-fixing antibodies. And many labs are probably not cross-matching patients in these cases. But if you are, then yes, probably a good thing to do would be to inactivate the serum to prevent cell lysis during your cross-match.
Okay, and do you have different cutoffs for samples separated using the regular method and samples separated using RosetSep and SetMate?
So based on our study, the cutoffs, where we compared the 2 cell isolation techniques, the cutoffs were virtually identical. I think there were 3 to 4 channel differences for T cell and B cells. It's probably just due to normal variability that you get with cross-match assay. So yes, they are slightly different, but not significantly.
Okay. Another question. Do you think that the RosetSep reagent can contribute to RBC contamination when using the SetMate tube?
I'm not sure. We tried using RosetSep reagent without SetMate tube, and we were not getting as much red cell contamination. So I think it's mostly the SetMate tube that does that. But our experience with this is limited because very quickly we've adopted the SetMate tube. So I don't have enough experience using RosetSep alone. But it's a great question.
Great. Okay, I'm just going to pass this over to Karina, our principal scientist.
Thanks. Yeah, we have actually tested this fairly extensively in-house, and in our hands we don't see a difference between the SetMate tube and just doing Ficol alone in terms of red blood cell contamination. There was also a question about why we increased or why the incubation for rosette sep is decreased from 20 minutes to 10 minutes in the SepMate tube, and so I can address that one as well. When you use SepMate, for whatever reason, we've just been able to decrease the incubation time down to 10 minutes. Now, I should say if you're dealing with older samples that tend to have lighter red blood cells, shall we say, or sometimes the red blood cells look a little bit less dense, you do need to spin the sample for longer. So you can still incubate for 10 minutes, but you might need to increase your spin time from 10 minutes up to 20. The really nice thing with the SepMate tube, though, is you can do your 10-minute spin and pull out your sample. And if it still looks red at that point, because it's an older sample, you can just pop it back into the centrifuge and spin it for another 10 minutes so you don't disturb the layers or anything. So I think that's actually a huge advantage to the tube. And that might also address some of the concerns about getting more red cell contamination with the SepMate tube.
Thanks, Karina. Another question for you, Rob. How many mils of whole blood do you start the prep with?
For the on a deceased donor, we typically use 4 tubes of whole blood, which I think has 6 mils. Yes, each ACD tube is 6 mils, so we would start with 24 mils. On a live donor, we would typically isolate from 2 tubes, so 12 mils of whole blood.
Great. There's just a question that's come through about whether you can use Ficol with the SepMate tube. Yes, you can use Ficol or any other standard density gradient media with the SepMate tube. The next question is about pronase concentration. Pronase concentration. Rob, can you comment on that?
Sure. Yeah, in our lab we use 4.7 Kunz units per mil. So rather than using the milligram per mil concentration, which, as you probably know, different pronase lots have slightly different activity, we determine our concentration based on the activity. So it's 4.7 Kunz units per mil. And we have, and again, I'm happy to share the protocol. We have kind of a graded amount of pronase that we add depending on how many cells we have. So we would perform a cell count before we pronase cells, and let's say if we have 10 million cells, we would use a mil. If we have 8 million cells, we would use 0.8 of a mil, and so on. So it's kind of a sliding scale depending on how many cells we have.
Right. Great. A question here about whether you've published the method or not.
We're working on it. Dr. Gable is actually keeps hounding me a little bit about this. I've been but yes, it's in preparation.
Great.
It should be out very soon.
Okay, we'll keep our eyes out for that. We've just got time for another 2 or 3 questions. So if you have them, please submit them now. The next one is, do you see a difference in MCF using frozen versus fresh cells?
That's an excellent question. Unfortunately, we do not use frozen cells. I know Dr. Campbell in Edmonton uses frozen cells, and she has a lot of experience with it, and I'm sure she would be very happy to answer questions about that and help him. And I think a center in Toledo, and Dr. Stepkovsky is one of the HLA directors there. They also use frozen cells, so this would be a good question to ask them.
Okay. Got another question here. I think I'll address this one to Karina. The question is, can the SepMate tube be used with a buffy coat to limit RBC contamination, and is a protocol available?
Yeah, that's a great question. the simple answer is yes, it can be used with a buffet coat, but it does depend on the red blood cell content that you have in the sample. If you're using, and it also depends on the size of tube that you're using. So in the larger tube, you need to have a packed red cell volume of 2 mils basically to ensure that your enriched layer is above the insert. And obviously this is much smaller in the 15 mil tube. Yeah, we do have some information available through our technical support team as well. So for the individual who's interested, we'll try and get a hold of you and send out a protocol that you can try.
Thank you, Karina. Okay. A question for you, Dr. Liwski. Can you comment on the cost effectiveness by using the RosetteSep and SepMate platform?
Cost effectiveness. Well, I mean, the isolation method obviously is going to be a little bit more expensive because you're using the RosetteSep, and the SepMate tube, I believe, is about $4 per tube. But you know, you're saving a lot of time. For example, if you're doing it on a deceased donor, if you save half an hour for the cell isolation technique, you know, you save half an hour of technologist time, and usually